Chisholm Maludu is a PhD candidate and associate lecturer at Anglia Ruskin. She's going to be talking about altering AD, ABC antibiotic sensitivity response with quadruplex DNA with genetic regulation towards applications against antimicrobial resistance. So we can see your screen. Great.
Great. Good. Hello, everyone. Good morning, and thanks for having me here today. I'll be taking you through my talk on quadruplexes and their role in altering efflux pump antibiotic sensitivity response. So we all know about the predicted death toll by 2050 as a result of AMR. Talked about this extensively, and the list by WHO having Acinetobacter baumannii in the critical category in need of R&D for alternate therapeutic approach. As a result, I have been studying the AD-ABC efflux pump genes as they have been observed to be overexpressed in multi-drug resistant strains, including strains resistant to the last resort antibiotic drug class carbapenem. My research focuses on the application of a tool, the quadruplex, within an area with imminent need of an uncommon approach, that's the antimicrobial resistance niche. So the significance of the efflux pumps in resistance was examined through a series of efflux inhibition assays with the use of carbon as an inhibitor. The observed decrease in the MIC with ertapenem in the presence of this inhibitor, that is the decrease in resistance also resulting in decrease in bacterial growth, confirms the contribution of the efflux pumps in the resistant mechanism of AD baumannii. So following on from here, there was a thought by proposing that a molecular tool quadruplexes having similar function of inhibiting the mechanism of the efflux pump genes in resistance without the cytotoxic effect of the inhibitors could pose a substantial option in developing a novel antimicrobial therapy for AMR. So quadruplexes have been shown to play significant roles in genetic regulation. This has been demonstrated extensively in the works of expert researchers from the University of Cambridge and UCL. These structures have confirmed roles in specific genes as seen here. Examples from literature also demonstrates the potential scope of functionality of quadruplexes in many areas within microbes. However, the full potential of these molecular tools, that's the quadruplexes, has yet to be fully unlocked. But why quadruplexes? Quadruplexes are four-stranded, but what are quadruplexes? Quadruplexes are four-stranded guanine-rich nucleotide structures from single strands of DNA or RNA sequences. These guanines self-assemble through the formation of hydrogen bonds and allow for the formation of a planar cyclic arrangement of 4 guanine molecules known as a guanine tetrad. So hydrogen bonds formed between the Watson Crick edge of one base and the host gene edge of the next. In the presence of metal cations such as sodium or potassium and interacting ligands, the folding of the nucleotide strand displays tetrads in stalk-like forms. For visualization, here is a schematic representation of a folded quadruplex structure. The balls represent the guanine bases interacting to form stacked tetrads connected by oligonucleotides, which form the loops. So now, why should quadruplexes be considered? The most prominent strategies have been the chemical engineering of antimicrobials. However, they have become less effective over time. Even more so, the time taken to develop resistance has shortened. To tackle this issue, I propose to counteract the resistance mechanism rather than develop further novel compounds which are short-lived. This aims to allow already existing antimicrobial compounds be useful again. So the planning theory was to find quadruplexes in a gene associated with resistance, in this case, identifying putative quadruplex sequences. These are sequences with the potential to form the quadruplex structures. So these were identified in AD-ABC efflux pump genes, and a similar quadruplex would be assembled externally and introduced in high concentrations into the bacterial cells, where they would competitively bind to quadruplex associated proteins. So where a quadruplex assembled in cell results in the upregulation of a gene, the introduction of an externally packaged quadruplex would in turn result in downregulation of the efflux pump and hence increase sensitivity to the antimicrobial agent. If this worked, we would expect to see reduction in growth of bacteria in the presence of both the antibiotic and the assembled quadruplex. So how was this theory tackled? The nucleotide sequence of the identified gene, that's the ADABC, was obtained from reference genome through NCBI database. A quadruplex predictive tool, that's the QGRS mapper, was then used to analyze for sequences with the potential to form quadruplexes using the formula as displayed here. In vitro experimental analysis involved the obtained putative sequences undergoing an assembly process through the instruction of monovalent metal cations, as well as water as a negative control by heat denaturation at specific high and low temperatures. The subsequent assembly products were further analyzed using UVV spectrophotometry, where a thermal difference spectrum was produced, allowing the observation of characteristic quadruplex spectra. Here we see TBA, which is thrombin binding aptamer that has been well established as a quadruplex forming sequence, and such was used as a positive control. So here is the TDS result from the TBA analysis. The characteristic quadruplex TDS can be observed to have a negative peak at 295 nanometer, as indicated with the region X. So analysis of all assembly products showed 5 sequences to demonstrate similar negative dip at 295, just as with the TBA, although they all have different strengths in the negative peak. So quadruplex further analysis to characterize these sequences was performed using DNase, as quadruplexes have demonstrated resistance to degradation by DNase. Therefore, we expect a maintenance of a gel band after DNase activity, which indicates the formation of a stable quadruplex structure. So here we do see the results of the DNase assay, where a double-stranded DNA sequence was used as a control to demonstrate effective DNase activity, and TBA was used to demonstrate quadruplex resistance to degradation. So as observed, the control effectively degraded and the TBA assembly product was unaffected. But also, although AD4, as we do see, shows significant degradation, which could be attributed to lower efficiency in the assembly product. Also, as it's seen in its peak in the TDS analysis, where it looks a bit weaker than the rest. AD2, 9, 11, and 13, however, did not demonstrate digestion by DNase, therefore, indicative that these sequences are able to form stable quadruplex structures in vitro. So here we do see, here we have a Acinetobacter baumannii, the NCTC 12156 type strain, which was treated with the assembled ADABC gene associated quadruplex. In this case, I picked AD9, and also in the presence of ertapenem, the antibiotics, at varying concentration. Interestingly, this resulted in approximately 50% decrease of the pathogen's MIC to ertapenem in comparison to when it was treated without the DNA being present. This finding is very exciting. However, further work is being carried out to elucidate this. In conclusion, as we all know, resistance of microbes to available therapeutics continues to gain an increased level of concern. The efflux pumps have been shown to contribute to these resistant mechanisms, and quadruplexes, having been involved in significant biological function, are also predicted to have an impact in the gene expression of the efflux pumps. Targeting the action of these physiologically active structures may inhibit resistance mechanism and restore the efficacy of antimicrobial chemotherapies, unlocking a new era for drug therapeutics. Furthermore, this study will act as a proof of principle with an expected ease of application in future development of novel therapeutics against AMR. I just want to say thank you to my supervisory team, also Carrie, who is here, and Chris also, and thank you very much.